My name is Toftgård.
My name is Teglund.
We will tell you about the background and implications of the study. Ever since we took part in the cloning of patch as the gene underlying Gorlin syndrome and development of basal cell carcinomas, hedgehog signaling has been a main interest in the lab. In a search for additional tumor suppressors in the pathway, we cloned human SUFU and showed that it was expressed in areas with active hedgehog signaling and that SUFU was a potent inhibitor of signaling and gene nuclear localization in cellular assays. This was the status when Stefan joined the lab.
So when I came to the lab, Rune presented the idea to knock out SUFU in the mouse. This sounded like a quite interesting idea. However, data from Drosophila genetics suggested that SUFU played a very minor role in the pathway, so we didn't know what to expect. However, when we got the first germline transmission and the F2 offspring, it became obvious that we were dealing with an embryonic lethal phenotype, to our surprise.
When my colleague Jessica Svärd and I started to analyze the embryos, not only did we discover that they are similar to patch1 knockout embryos, in addition the SUFU heterozygotes by time develop Gorlin syndrome-like features, and this includes basaloid changes of the skin and jaw keratocysts. We now know that SUFU germline mutations in human also can cause Gorlin syndrome.
So, the fact that the absence of SUFU causes a constitutive pathway activation really convinced us that SUFU played a major role in mammals.
Since this study was published, we have learned much more about SUFU. However, a key remaining question is how the hedgehog signal is transduced from smoothin to SUFU glee and how glee repression is relieved. This may involve post-transcriptional modification of SUFU linked to a conformational change, as well as interaction with additional partner proteins.